Characteristics of Dicot and Monocot stem and root. Materials required: cover slips, filter paper, glass slide, fresh material of stem, distilled water, saffron in solution, glycerine, brush, needle, dropper, watch glass, and a compound microscope. Procedure: take a blade and a stem to be sectioned. Hold the stem to be sectioned horizontally between the thumb and the first finger of your left hand. Cut sections of the material quickly using the blade. Transfer the sections into a watch glass containing water. Using a dropper, take some distilled water and pour it into a clean glass slide. Using a brush, select a thin, uniform and complete section and place it on the glass slide. Using a dropper, take some saffron in solution. Pour a few drops of saffron in solution over the section and allow it to stand for 2 minutes. Remove the extra stain using a filter paper. Take a few drops of glycerine in a dropper and pour 2 to 3 drops on the slide. Take a cover slip and place it gently on the section with the aid of a needle. Place the glass slide on the stage of the compound microscope. Observe under the microscope. Observation: TS of Dicot stem. We can observe the TS of a Dicot stem through the microscope. The Dicot stem has an outermost layer made up of parenchyma cells known as epidermis. The epidermis is covered with cuticle towards the outside. Below the epidermis lies the hypodermis, and it is made up of a few layers of collenchyma cells. Several layers of parenchyma cells that lie below the hypodermis is known as cortex. Endodermis is a single layer of parenchymatous cells that separates vascular tissue from the cortex. The vascular bundles are wedge-shaped and arranged in a ring. Each vascular bundle is conjoined, that is, xylem and phloem occurs in the same bundle. Collateral, that is, phloem lies towards the outer side and xylem occurs towards the inner side. And open, that is, cambium is present between phloem and xylem. Medullary rays are made up of parenchymatous cells between the vascular bundles. Features of identification: presence of multicellular hair on the epidermis, presence of collenchymatous hypodermis. Vascular bundles are wedge-shaped and are arranged in a ring. Each vascular bundle is conjoined, collateral and open. TS of monocot stem. We can observe the TS of a monocot stem through the microscope. The monocot stem has an outermost layer made up of parenchyma cells known as epidermis. Below the epidermis lies the hypodermis and is made up of a few layers of sclerenchymatous cells. Ground tissue is composed of parenchymatous tissue and extends from hypodermis to the center. Endodermis is a single layer of parenchymatous cells that separates vascular tissue from cortex. The vascular bundles are numerous and scattered in the ground tissue. Each VB is oval in shape and possesses a sclerenchymatous sheath. Each vascular bundle is conjoined, collateral and closed. Features of identification: absence of short hair, Presence of sclerenchymatous hypodermis. Ground tissue is undifferentiated. Vascular bundles are oval in shape, numerous and scattered in the ground tissue. Each VB is conjoined, collateral and closed. Xylem vessels are arranged in a V-shaped manner. TS of dicot root. We can observe the TS of a dicot root through the microscope. Dicot root has a single outermost layer of thin-walled, compactly arranged cells called epiblema. Some cells of epiblema bear thin-walled, tubular outgrowths called root hairs. Cortex lies below epiblema and is made up of many layers of thin-walled parenchyma cells. Endodermis is a single layer of barrel-shaped cells. It separates the cortex from vascular tissues. The cells of endodermis possess band of thickening called Casparian band. Pericycle is a single layer of thin-walled cells below the epidermis. Vascular bundles are present 2 to 6 alternatively arranged bundles of xylem and phloem called radial bundles. The center of the root is occupied by pith made up of parenchyma cells. Features of identification. Presence of unicellular hair on the epidermis. Vascular bundles are radial. Xylem and phloem are less than 6. Xylem is exarch. Pith is either reduced or absent. TS of monocot root. We can observe the TS of a monocot root through the microscope. Monocot root has a single, outermost layer of thin-walled, compactly arranged cells called epiblema. Some epiblema cells give rise to unicellular root hairs. Cortex lies between epiblema and is made up of many layers of thin-walled parenchyma cells. Endodermis is a single layer of barrel-shaped cells. It separates the cortex from vascular tissues. Pericycle is a single layer of thin-walled cells below the epidermis. Vascular bundles are present many, 8 or more, alternatively arranged bundles of xylem and phloem called radial bundles. The center of the root is occupied by well-developed pith made up of parenchyma cells. Features of identification. Presence of unicellular hair on the epidermis. Vascular bundles are radial. Xylem and phloem are more than 6. Xylem is exarch. Large pith is present in the center.